Okay, hey guys, I apologize for not having a live recording from class. When I got to my Zoom dashboard this afternoon, everything was all wonky and what was supposed to automatically be there was not automatically there. So I think I have reset it so that it will be functioning properly for next week, but I wanted to go ahead and go over our lesson again anyway, so make sure you had slides to study for your Unit 1 test. Occasionally we will have a tech failure like this, and usually I don't re-record the entire lesson because this was the first lesson, though I felt it was necessary to do so. So the first thing we did was go over our house rules. These are in your preface. This was just a reminder of what the expectations are for being in our home, and you can review those in your preface as well. The next thing we did was open up our binder to our preface. I think it was page 5, and made the following edits that I did not catch before I printed them. Your final project paper upload is due 5/17/26, and your presentation will be 5/19/26. So go ahead and fix those if you would. All right, moving on to the lesson proper. What is science? Science is an organized way of gathering and analyzing evidence about the natural world. We are in natural sciences. The formal sciences are things like logic, statistics, mathematics. Natural sciences are about the natural world. What is science and what are the goals of science? Okay, so the goals of science are to investigate and understand nature, explain events in nature, and to use those explanations to make predictions. So today I use the example of dropping a little ball of wadded up paper to the floor. If I do that several times, I've investigated that every time I let go of that little ball of paper, it falls straight to the floor. So now I need to try and explain that. Well, part of what we have come up with to explain that is a theory based on gravity that says that gravity is the attractive force between any two bodies, and that a body with a greater mass will have a greater pull on a body with less mass. So while both the ball of paper and the earth are pulling towards one another, the earth's pull is going to be greater than that of the ball of paper, so the paper is going to appear to fall to the earth. And then we can use that explanation to make a prediction. If we do it again, if we drop the little ball of paper, it's going to fall again and again and again. We talked about how scientific method is an organized process for studying things, how it involves observations, questioning things, forming hypotheses, and then making predictions, and how it is not always a perfectly straight line because sometimes you need to revise a hypothesis, or sometimes you need to change your methods because it's not working well. Sometimes your hypothesis is unsupported and you have to start all over with some other new hypothesis. Now, this is something that will show up on every midterm and final test that I give, and it is the 3 experimental values, and they are as follows. Now, an independent variable, experimental variables. I think I said values. Goodness, I'm tired. An independent variable is what you change in an experiment. You will sometimes hear this called the manipulated variable because you manipulated it, you changed it. Your dependent variable is what changes based on what you change. And you will sometimes hear this called the responding variable, and that is because it is responding to the change you made. And then your control is what you keep the same. So I gave the example again of having a headache. If I have a terrible headache, I want to change something to make that headache go away. So I take a Tylenol and the headache goes away. Okay. The independent variable was the addition of Tylenol. The dependent variable was the alleviation of the headache. But you can't say for sure that's what caused it until you repeat your experiment multiple times. And if as long as you keep everything else the same, though, it's controlled. But if I take a Tylenol, put a cold compress on my head, go to bed in a dark room, and my headache goes away, I have now changed far too many things. This is not a controlled experiment. We changed more than one variable. And in this case, we can't even begin to make a statement about what caused the change, what changed based on what we changed, what caused the headache to go away, because we had way too many variables involved. This also is on every midterm and final that I give, and it's about hypotheses. A hypothesis is an idea that can be tested. It can either be supported or rejected, or accepted or rejected, or accepted and not or accepted or not supported, however you want to phrase it. But we never say true or false or proven or unproven because that's not good scientific language. And the outcome of your testing must be repeatable, or you can also say reproducible. Also on every midterm and final I ever give is the difference between a scientific theory versus a scientific law. A scientific law is what happens, a fact, A scientific theory is why it happens in explanation. So if we go back to our example of dropping the little ball of wadded up paper to the floor, if I open my hand, the paper will fall. If I open my hand, the paper will fall. That is what is happening. It is a fact. It is a law. Gravity is a law, but also a theory. The theory of gravity is explaining why, and that has to do with the definition I gave about gravity being the attractive force between any two bodies, and that bodies with a larger mass will have a greater attractive force than bodies with a lesser mass. So now we have both a theory and a law involving gravity. Some things are both, some things are not both theories and laws. Some things are one or the other. Biases. So biases, in science, a bias means a tendency to favor one thing over another, which can make the results unfair or inaccurate. It's like having a favorite sports team and always thinking they're the best even when they lose. In science, it can happen when researchers unintentionally lean towards certain results or use methods that favor one outcome over another. We are human beings. We are emotional creatures. We are not these infallible, rigid, objective creatures. We aren't computers. We aren't the other types of tools that we use to measure quantitative data. That's not who we are. So we can't change that, but we have to be very aware that we are being as objective and non-biased as possible when we are working with science, because inevitably our biases are going to creep in. And while we'll never be perfect, we can always make an effort to be aware of those biases at all times. So life science is biology, and we talk about how we use a lot of Greek and Latin roots and how when we become familiar with them, they'll be very useful because they can help you decode a word that you've maybe never seen before. So in this case, ology means the study of, and bio means life. So the study of life is biology. And we'll see some other ologies in a minute. Gathering data. Observations. We tend to think of observations about what did you see? Like if a police officer, if you witnessed an accident, they'd say, what'd you observe? And you would probably tell them a whole bunch of visual information. But that's because we tend to lead with vision as our strongest, most reliable sense. We get the most input, those of us who have typical vision. But observations are made with your five senses: taste, touch, sight, smell, and sound. But we do have a sixth sense in there that isn't often included, and that is the sense of proprioception that has to do with where your body is in space and whether it is moving or still. So this is like how you can sit in a car, close your eyes, and still tell if you are turning right or left. It's also how you know that when you're sitting in a chair, you're not upside down. So that is another sense to be aware of at all times. But observations can be either quantitative or qualitative. Qualitative data is descriptive, non-numerical, focuses on qualities, characteristics, and experiences. So like at the bottom, was it cold or hot? How did it smell? How did it feel? What color was it? Was it a high-pitched, a low-pitched sound, loud or soft? Whereas quantitative data, on the other hand, is measurable, numerical, focuses on quantities, amounts, and frequencies. So things like what was the angle? What was the temperature? Not was it warm or hot? What was the temperature? What was the time? What was the wind speed? Things of that nature. And so essentially your qualitative is why or how, while your quantitative is how many, how much, or how often. So we can use this little tarantula as our example. Saying the tarantula has 8 legs is a quantitative observation. You can count them. It deals with something numerical. But saying that the tarantula feels fuzzy is a qualitative observation. It has to do with how we experienced the tarantula. So an inference is made based on prior knowledge and experience. So let's insert some prior knowledge here with what we know about that feeling fuzzy 8 legs. So only mammals have true hair. Most people know that, most people at your age at least. So the fuzzy hair on a tarantula must not really be hair. All right, that's prior knowledge. Mammals are warm-blooded. We're going to use a better word for that later. And hair keeps them warm. Tarantulas aren't warm-blooded, so their hair must not function as an insulation. So what is the hair, quote unquote, on the tarantula's legs for? It has to do with helping it with sensory input. So it's not hair in a typical sense. And we know that because we have prior knowledge. So that's an inference. Predictions. And now the weather. It will be sunny unless it rains. And that is how we often feel these meteorologists talk to us, right? So we can use our observations and our inferences to analyze our data, and we can make predictions. Now, we played a little classifying game. We have some figures here, symbols, and we're going to classify them based on their qualities and characteristics. So the first way we classified them, everyone agreed that these were classified by color: yellow, blue, red, and green. And the next way we classified them, we decided the things on the top were like animals, the things in the middle were machines, and the things on the bottom were plants. That's how we classified them in that case. And in this case, we all agreed that they were classified by solid figures or outline figures. And in this example, we all agreed that the items are classified by means of locomotion. The apple and acorn, I cannot speak, I've been talking all day, do not move, so they have no means of locomotion. The cyclist uses wheels, whereas the plane, butterfly, and bats use flight, and then the bear, beaver, and caterpillar use legs or creep or walk. And then lastly, we all agreed that the top figures were mammals, the middle were insects. The airplane was off to the side, kind of even, because it's not a living thing. And then at the bottom, we had plants or fruits, or however you want to put it. We also noted that the man on the bicycle could be categorized when you observe him as a man or as a bicycle. So it kind of depends on how you interpret that image based on how you're based on how you are categorize it. So he could go either way. We talked about models and how models are used in science very often to help you visualize very small things that are too hard to see, like cells, or very large things like the solar system, which you can't just see with your eyes on your own or observe in any other way. We also talked about how models can be anything from graphs to globes, and we will be using some models this year for those little tiny things as well that you can't see because they're so little, but also for processes like photosynthesis. We can model those processes. We talked about how there are many different careers in biology, and here are all the ologies: zoology, study of animals; mycology, study of fungus; microbiology, study of very small living things; immunology, study of immune system. So once you start to understand your the Greek and Latin that shows up in scientific words all the time, you will become pretty familiar with it, and it will help you to decode words that maybe you haven't even seen before, like mycology. Don't know if you've seen that one before. So it's important in science to be curious, honest, open-minded, skeptical, and creative. Curiosity and creativity kind of go together. It is curiosity and creativity that has led to some of the greatest scientific discoveries of all time. Honesty and open-mindedness, this is where your bias comes in. Be honest when reporting data and be open-minded to the data not being what you expected it to be. Skepticism seems to kind of go opposite open-mindedness, but not really. Skepticism is more about always considering your source, never taking anything at face value. When you're considering the source of your data, keep in mind that, like, your social media influencers, they aren't so much a good source of data. So that's what we mean about being skeptical. And yes, we caught the technology was spelled wrong, but technology doesn't have to be computers or anything. It can be a pencil, a sneaker, that's technology too. Engineering and science is important. This helps us build and bring to life some of our greatest ideas. And then here are some people who've given some awesome impacts to science over the years. And this year in particular, we'll be dealing with Darwin and Mendel. A couple of these other guys might come up. So there is our biology. Concepts in biology. Living things are made of cells, and DNA carries a universal genetic code for all living things. All living things grow and develop and reproduce. We talked about how some living things tend to sort of freeze their growth and development. For example, cicadas. They can bury themselves in the ground for years and they are sort of in a suspended development, but they do grow and develop when they come up out of the ground and make all the lovely noise that we hear, and then they mate and then they die. There was also some discussion of frozen embryos. That's another example of how you can kind of arrest development. But growth and development and reproduction are still possible with all of these things. Therefore, they are living things. So even like an embryo that's been suspended in a cryo type environment is still a living thing because it has the potential to grow and develop and reproduce. So concepts in biology. Taken as a group, living things evolve. Branching from a common ancestor, form is a reflection of function. So your ear is shaped the way it is because it helps you collect sound. That's why it sticks off your head. But if you cup your hand behind your ear to hear somebody better, basically what you're doing is making that part of your body that is meant for capturing sound even bigger. So you kind of amplify the sound. Now, why our ears aren't as big as they are when our hands behind them? Probably because we don't need that much sensory input. So everything has a function, and the form is based on the function. And the evolution of living things is less of a straight line, and it looks a little more kind of like this crazy image here to the right of the screen. Metabolism is the process by which living things turn matter into energy. You hear about some people having a fast or a slow metabolism, like whether they gain weight easily or whether it's hard for them to gain weight, is usually how you hear people use that word. And then homeostasis is the tendency toward a relatively stable equilibrium between interdependent elements. And that is things like osmolality, like maintaining a equal amount of salt on one side of a membrane versus the other side, balancing out. And in chemistry, we just call this equilibrium, but it really is more complicated in biology, so we call it homeostasis. Now, schnapps. We call this schnapps in biology. Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur are the elements most common in all living things, from the smallest bacterium to the biggest blue whale. These are the elements that you're going to find in those living things. So cute things and not so cute things, tiny things, flowers that are really weird, gecko creatures, mushrooms, grapes. All of these things contain these same elements in greater quantity than any other element. And we can't forget the water bear there, little tardigrades. Super cute. Concepts in biology, interdependence exists in nature. Food webs are a better example than food chains. And I think you can understand why. A food chain is basically the bird eats the seed, the cat eats the bird, the dog eats the cat, but it doesn't show that there is an interdependence. The nature of matter. Matter is anything that has mass and takes up space, and it is composed of atoms. And in our atom, of course, you have your protons and neutrons in the nucleus and your electrons spinning around in the cloud outside. Remember that word is nucleus, not nucleus. That is also why it is nuclear, not nuclear. That is one I disown people for. The nature of matter isotopes contain a different number of neutrons. So in an atom that is an isotope, it has an additional neutron or maybe one less neutron. So that changes the mass of the atom, but it does not impact how the atom interacts with other atoms because that has to do with its charge. That's when you're dealing with ions. But isotopes have to do with the mass of an atom, and it has to do with a different number of neutrons in the nucleus. Two or more elements combined in different proportions form a compound. We don't say molecule here for a reason. A molecule can be defined as covalently bonded, but it can also be defined as two of the same atoms together. So the example I gave today was oxygen. Oxygen does not like to be alone. He always has a friend. So oxygen always goes out in pairs. So it's always O2, never just O by himself. That is a molecule, whereas a compound involves two different elements. So we have ionic bonds where one is giving an electron and the other is taking it. And then we have covalent bonds where they are sharing electrons. This is just kind of a review of some basic chemistry. For people who did chem last year, it's a good review. For people who never touched it, it's just kind of a nice base that will help you through the biology course. Water. Water has some very important properties that make it so essential here on Earth. It is polar, which means it has a more positive charge towards one end of the molecule and a more negative charge towards the other end of the molecule. It is attracted to other molecules by adhesion. You see that down here in the bottom right-hand corner, adhesion, attracted to other molecules. It has a high heat capacity. So a huge amount of heat is stored. Water's heat capacity stores a lot of heat, which is why it is difficult to boil. It's also difficult to freeze. It takes time, right? And that is because it has a high heat capacity. Hydrogen bonds bind water to water. So you have polar bonds, and the molecule itself is covalently bonded, and you've got hydrogen and oxygen bonded to each other, and that's a pretty strong bond. But then you have hydrogen bonds that bond one water molecule to another water molecule. And that's how the reason for things such as capillary action, and surface tension. And capillary action is like if you touch a paper towel to a drop of water on a counter, you're going to watch that water move up into the paper towel. And that's because the water molecules are attracted to one another through hydrogen bonds. And then also you have surface tension, which you can see anytime you see like a little water droplet, it's rounded on the top. It's also why little bugs can run across the surface of water sometimes. pH and buffers. So here's a pH scale for you. Things on the lower end of the scale are more acidic, and things on the higher end of the scale are more basic. People tend to think of, oh, acid burns. Well, so do bases. Strong bases will burn too. Look at that. Drain cleaner is there at 14. You don't want to get that on you. 7 is neutral, and that is the pH of water. So 0 to 7 is acidic, 7 to 14 is basic. And buffers. Buffers are chemicals that you add to reactions very often. Sometimes they're there naturally, but they resist a strong change in pH. You have buffers in your blood and in other parts of your body that help maintain a steady pH. So when chemical reactions are ongoing, there aren't giant shifts in pH. Carbon compounds are carbohydrates and lipids. Carbohydrates you can think of as like sugars and starches. Lipids you can think of as oils and fats. Carbohydrates are short-term energy storage. Lipids are long-term energy storage. Carbohydrates are going to have more osmotic pressure. So if you have a membrane with a higher sugar concentration on one side, it's likely to balance through that membrane. Whereas if you had a membrane with a higher fat concentration on the other side, it's not going to balance across anywhere near as readily. So we'll talk more about these as we go throughout the year, but these are just basic understanding of carbohydrates and lipids. So carbohydrates are naturally occurring compounds made or a derivative of such a compound made up of carbon, hydrogen, and oxygen. They are the most widespread organic substances. They play a vital role in all life. Lipids, in particular, a great example of a lipid is in a phospholipid bilayer. This is basically a cell membrane. You've got a hydrophilic and hydrophobic end, or a water-loving and a water-hating end. And basically, you see that little one that's all by himself up there at the top. You've got kind of like a fatty lipid end that is attracted to lipids on the outside, and then those where the tails all touch, some are inverted and some are upright, that would be all water loving. So this helps create this bilayer, and there's a good reason for this. And when we talk about cellular transport, we'll discuss it more, but sometimes things can pass in and out of that membrane without a lot of energy. Sometimes you need channels, like there are actual structures that make channels, and sometimes those channels act as pumps where you actually have like a gradient on one side or the other, and there's an active transport going on. It's pretty cool. So lipids are any class of organic compounds that are fatty acids or their derivatives. They are non-polar, which means they will not mix with water because water is polar and water and oil don't mix. And they include things like natural oils, waxes, and steroids. Nucleic acids, amino acids, and proteins. Nucleic acids are found in DNA, and they code for amino acids. Your DNA code will create a code for producing certain amino acids, and then the amino acids will join together to form a chain or a protein. So these would be your nucleic acids and whatever order they are in. And we're going to go over this. We're going to do some activities where you actually like decode secret messages and things. But as these code these work in groups of three, basically, and a group of three of these will tell your body to produce a specific amino acid. And then once that amino acid is formed, another one will be formed behind it, and then another one behind that, and it makes a chain. Proteins don't behave as a chain, though. They actually fold up like origami in a very specific shape, and it is that shape that is essential in how the protein functions. So proteins are bonded with what we call peptide bonds. The sequence of amino acids in a protein is defined by a gene and encoded in the genetic code. Although this genetic code specifies only 20 standard amino acids, the residues in protein are often chemically altered in a post-translational modification, either before the protein can function in the cell or as part of the control mechanism. So though we only produce 20, some of them get changed before they start being active. Proteins can also work together to achieve a particular function, and they often associate to form stable complexes, like other biological macromolecule, large molecule, macromolecule, such as polysaccharides, many, saccharide, sugar, a long chain of sugars, and nucleic acids. Proteins are essential parts of all living organisms and participate in every process within cells. Many proteins are enzymes that catalyze biochemical reactions and are vital to metabolism. Enzymes are catalysts are anything that lower the activation energy of a chemical reaction so that it takes place more quickly and efficiently. And enzymes are catalysts found in nature. So today in lab, the particular enzyme we were testing is called catalase, and it exists in the liver, and it helps the liver break down hydrogen peroxide into harmless water and oxygen. So that's what we did today in lab is we took advantage of the fact that that enzyme produces oxygen, which makes bubbles. So it's something you can see. And we measured how different temperatures affect the performance of that enzyme, catalase. Other proteins have structural and mechanical functions, such as the proteins in the cytoskeleton of a cell, which form this scaffolding in there that kind of maintains the cell's shape. And proteins are important in cell signaling, immune responses, cell adhesion, and the cell cycle. Proteins do everything, you guys. A protein is also a necessary component of our diet. Since animals cannot synthesize all the amino acids, they must obtain essential amino acids from food. We can create 20 amino acids, but there are more than 20, and some of them we have to get from what we eat. This is why people that have more restricted diets, like they don't eat meats, for example, or dairy, sometimes have to get their proteins from other sources. Well, they do have to always get their proteins from other sources, but they have to be sure that they are eating the right kinds of foods to get the right kinds of amino acids that our bodies cannot produce on its own. Though through the process of digestion, animals break down ingested protein into free amino acids that can be used for protein synthesis. Chemical reactions and enzymes. A chemical reaction changes one set of chemicals into another set of chemicals. So this is an example of a chemical reaction at the top of your screen. You have reactants yield products. So on the left, we call those reactants, and on the right, we call them products. And this particular reaction, where you have glucose, C6H12O6, and oxygen, 6 of those, there's O2, remember I said he's never alone, and 6 carbon dioxides, and 6 waters, this is aerobic respiration. So that is the process that goes on inside your cells. Chemical reactions and enzymes. Enzymes are catalysts which occur in nature, which is what we said. And catalysts lower the activation energy for a chemical reaction to begin. So if you look at this graphic here, you'll see right here, this red line is how much energy you would need for a reaction to reach activation energy, which is right here, at which point the reaction will continue. But when you add an enzyme, it lowers that activation energy. So that reaction will happen more efficiently and more quickly with less energy. Here are your Unit 1 study topics. So be sure that you go through these and study for your Unit 1 test. And then, oops, sorry. And I will see you next week.